All right, ladies and gentlemen, welcome back. So today we are going to wrap up our discussion on the regulation of the cell cycle by moving on to external signals. So things that cells are going to get from their environment or from neighboring cells to sort of prompt whether or not they should be dividing. So external signals can come in a couple of different ways, one of which can be these molecular signals called growth factors. And these are proteins that are released by certain types of cells in multicellular organisms that actually stimulate other cells around them to divide. So when those growth factors get released into their environment, they will go and connect to receptors on neighboring cells, which can then trigger the division of that neighboring cell. We see this commonly in fibroblasts. So these are common cells that are found in connective tissue, and their job really is to synthesize the extracellular matrix, the collagen, so sort of what holds all of those materials together in a cell. And that's kind of what you're looking at here in this figure, is just our understanding of how this regulation ends up working. So what they did was they took a sample of connective tissue, cut it up into small pieces within a petri dish, um, and then utilized enzymes to digest that extracellular matrix, and they resulted with just those free fibroblasts. So they then cultured them and looked at whether or not the presence of this molecule called PDGF, platelet-driven growth factors, had any impact on whether or not those cells would divide and then ultimately produce a tissue layer. And what they ended up finding is that whenever that PDGF, that platelet-driven growth factor, was in fact present in the culture, they were in fact able to stimulate those fibroblasts to produce that extracellular matrix, stimulate each other to start dividing, and ended up producing connective tissue within that culture. So we see that particular PDGF very prevalently when we look at wounds. So when we see neighboring cells, especially in things like your skin and your sort of connective tissue, excuse me, underneath that layer, producing these PDGFs, these growth factors, they then stimulate other cells within the neighboring environment to also start dividing, and that ends up being able to heal up that wound, seal up that wound, and then we'll sort of take care of that scar tissue and all that kind of other things, if possible, by additional cellular division. So this is just kind of what they were looking at under the microscope in what was going on in those platelets, or excuse me, in those plates. And here are those fibroblasts. They start producing that those growth factors, stimulating those other cells, and we can see that they start multiplying from there, which is pretty cool. Okay, so other things that we have been able to witness, identify in tissue culture, and as a result, be able to apply to what's going on in your cells, is these two things here, density dependent inhibition and anchorage dependence. And both of these are, just like everything else, molecularly controlled, um, but they're interesting when we look at cells and tissues. They should be exhibiting these two behaviors based on the molecules that they produce and the molecules that are typically in their environment. So what are these? When we talk about density dependent inhibition, to inhibit something means to stop it. So when we talk about cell division, the cell cycle should be inhibited when there's a large density of cells surrounding each other. So if I put cells into a culture, right, if I only put a couple of them in there, they should be able to divide until they start running into each other in a flat sheet. All right, so all of your tissues generally are in flat sheets. And if I run into the next cell as I'm dividing, I should stop dividing. Okay. The other idea here is the anchorage dependence. So cells need to be attached to a substratum, a flat surface. In your body, it's your extracellular matrix. When we do this in tissue cultures, it's an artificial matrix that we create within the culture tube. So the cells need to be not only capable of attaching to that flat surface, and then they're allowed to divide. But then once they divide far enough that they are now squished up against other cells, the division should stop there. Why are we talking about this? Well, because we're going to spend a little bit of time talking about some properties of cancer here. And when we look at cancerous cells, by definition, cancer is uncontrolled or unregulated cell division. And when we look at the way cancer cells grow in tissue cultures, we see that they're ignoring all sorts of internal and external signals. And some of that relates to this idea of the fact that they don't in- exhibit this density dependent inhibition, and or they don't display this anchorage dependence. And so that's where we can talk about that tumor formation coming from. So as those cells sort of start massing and growing on top of each other because they're not listening to that density dependent inhibition, they're not stopping their growth when they do hit another neighboring cell, that's where you get those massive balls of cells called tumors. And that anchorage dependence, when they are able to just sort of free float in tissue fluid in your blood and continue to divide when they shouldn't be, that's where we can get things like metastasizing. So cancer that starts spreading to other parts of your body away from its original tumor because it's now ignoring that anchorage dependence right along with the density dependent inhibition. So your cancer cells in this case don't respond normally to your body's control mechanisms, right? They're not listening to those growth factors. There may be genetic mutations that we'll talk actually a little bit about later this year. Um, Here we're just talking about the fact that something is going on internally and or externally, and oftentimes a little of both, that are causing them to ignore all of the traditional mechanisms that your cells have evolved to only divide when necessary. And so cancer cells can make their own growth factors. They can ignore whether or not growth factors are present and still divide. Um, they can, as a result, lack that density dependent inhibition, lack that anchorage dependence, and that's where we get those masses of tumors that come from traditional cancer diagnoses. We're looking for those tumors. Where are they? They'll often go in and biopsy them to see whether or not they're benign or malignant. Um, and then they start worrying about whether or not those cancer cells are capable of spreading. So we're looking at transformation, right? A normal cell, something has happened to it in the process of its probable required cell division, that has now converted it into a cancer cell. And that division can be just a simple mistake in DNA replication. It can be a causative mutation from some sort of environmental factor, something that you have in your diet, something that's in your environment like radiation or other chemicals, and even viral infections can have connections to the transformation of certain cells and cause them to become cancerous. So when that initial tumor starts building, we now have a situation where those cells are lacking that density dependent inhibition. They're growing on top of each other, they're massing together, rather than staying in the thin sheets that they're supposed to grow in as tissues. And so then we start questioning, once that tumor has been found, it becomes biopsied by medical professionals, and we're looking to see whether or not that tumor is benign or malignant. And so those benign tumors are often sort of misnomered to be non-cancerous. It doesn't necessarily mean that, but it just means that they are lacking enough um, mutated instructions that typically the tumor will remain at its original site. And so you'll often come across it as being non-cancerous, and they're not worried about the tumor cell spreading. So we're lacking the density dependent inhibition there, but not necessarily the anchorage dependence. And those tumors tend to be easier to treat because we can go in, cut them out, and that should theoretically be the end of it. When we look at malignant tumors, however, these are the tumors that we are particularly concerned about as far as cancer and them spreading. So malignant tumors not only have the markers for ignoring that density-dependent inhibition, but they also have markers for the potential, at least, to ignore the anchorage dependence as well. And when that happens, it's possible for some of those pieces of that tumor to sort of end up in your lymphatic vessels, your blood vessels, and start transporting themselves throughout your body and being capable of dividing all at the same time. So when they sort of get lodged somewhere else, say from breast tissue into your liver or into your stomach or into your brain, and start forming those secondary tumors, that's when we talk about that metastacism. So those cancer cells have now left their original tumor, traveled elsewhere, and started forming secondary tumors. The whole goal is to hopefully catch those cancer cells, get rid of all of those malignant tumor cells early, and hopefully prevent that metastacism from happening in the first place. But it does become considerably trickier when we do talk malignant tumors to ensure that we have, in fact, gotten all of the cancer cells and that those secondary tumors don't show up. So here you can just kind of see some breast scans. So here's sort of a normal breast tissue, and here is clearly one that's got quite the honkin' tumor going on there, and that's where we start looking for concerns as to whether or not that happens to be malignant or benign.
Hi, it's Mr. Anderson, and welcome to Biology Essentials Video 28. This is on the cell cycle, mitosis, and meiosis. In other words, how we go from one cell to all the cells, the trillions of cells inside our body. Meiosis is important because that's how we make sex cells. Now, let me digress a little bit. This is a whiptail lizard from the desert Southwest, and what's interesting about this is that it's a female lizard, and when it wants to make more lizards, it will simply use mitosis to make an exact copy of a cell inside its body. It's called parthenogenesis, or virgin birth, and it will make a brand new number of baby lizards, and they're all females. So they don't have males. It's rare to not have males. It's rare to not have meiosis, and the reason why is that all the whiptail lizards are genetically the same, but it works. And if you live in a fairly stable environment, it tends to work. Now, we're not going to go into the specifics of mitosis and meiosis. You can look, I've got videos on each of those that talks about the different phases. What I want to talk about this is, in general, what do mitosis and meiosis do, and how does the cell cycle work, and how is it controlled? And so a diploid cell is going to be a typical human cell, or a typical cell inside an organism. It's going to be 2N, and what that means is it has two complete sets of chromosomes. So for example, in humans, 2N equals 46. That means we have 46 pair of chromosomes. And so the goal of the cell cycle in mitosis is to make a copy of that cell. In other words, to make a diploid cell. That diploid cell can enter into the cell cycle again, make more cells and more cells and more cells. And so the way that we make new cells in our body or replace cells that are damaged is mitosis. In meiosis, we're going to take a normal diploid cell, and we're actually going to make a haploid cell. We're going to make sperm and egg. And so in humans, N now equals 23. Now, if we were to just stop there, we wouldn't have diploid cells anymore. But fertilization, where egg meets sperm, is going to combine those two cells to make a diploid cell. And now that diploid cell can enter into the cell cycle again. And so we've got kind of two loops. We've got the mitosis loop and the meiosis loop. Mitosis is used to make all the cells in our body. Meiosis just makes gametes, or sex cells. I also in this video want to talk about how we control the creation of diploid cells. So it may be better to come from here. And how we use cyclins. An example of that would be the mitosis promoting factor to control the cell cycle and where it is and where it's headed next. Now, if you're talking about cell cycle, the best place to watch is start is with videos of actually cells dividing. And so this is a cell undergoing division. So we start with one cell, and it makes an exact copy of itself. Now, when you're watching it, let me go back a second. On these first two videos, what you don't see is everything that actually happens before the cell makes a copy of itself. In other words, before the cell is actually able to divide, it has to duplicate all of the DNA and all the machinery of the cell. This last one, we're looking actually inside the cell itself. And so the cell division actually has two parts to it. Part one is going to be the division of the nucleus. And we call that, in general, mitosis. And then after we've divided the nucleus, you'll see the chromosomes actually separate here. Then we actually have the division of the rest of it, and that's called cytokinesis. Cytokinesis is the break apart of all the other parts of the cell. So the machinery of the cell, the mitochondria, cytoplasm, things like that. And so let me see if I can start that up. So first of all, there we go. First, we have the division of the nucleus. You can see the chromosomes pulling apart, and then you have cytokinesis, or the division of the cell itself. And so all mitosis is one cell forming two cells, and those two cells are identical to that first cell. And that's how we go from that first fertilized egg inside us to the trillions of cells that we have inside an adult body. So when you're replacing cells in your body, you're doing it through mitosis. So let's look at the cell cycle. And so what happens is you'll have a cell enter into this. So a cell is going to look like this. It's going to enter it into the cell cycle as one cell. Let me make a little better arrow. And then it's going to eventually exit out as two cells. Each of those cells, remember, could go back into the cell cycle. And so this is how we make all the cells in our body. Now, I've heard that a lot of the dust in a room actually are dead skin cells. And this is what skin looks like. So skin is going to be created new cells, and then they're going to migrate up to the surface, and then we're eventually going to lose skin cells at the top. But we keep replacing those cells, and to do that we use the cell cycle. So let's look at the parts of the cell cycle. If we start first with that cell entering in, the first thing that it'll do is it'll actually enter into the G1 phase. G1 phase, the cell is actually going to grow. It's going to get bigger and bigger and bigger and bigger. We then enter into the S phase. During the S phase, we're going to actually, using DNA replication, we're going to copy all of the DNA inside a cell. It then goes into the G2, or the growth 2 phase, where it continues to grow and gets ready for division of the actual cell. And so G1, S, and G2 are all part of what's called interphase. And if you look at a cell, it's generally in interphase. It's growing, it's copying its DNA, it's growing, or maybe it's just working. It's doing what a normal cell does. And so most of the life of a cell is in interphase. It's in these three, G1, S, and G2. And the actual copying of the nucleus and copying of the cell, this mitotic phase, is actually going to be really, really small. If it never divides again, it'll actually stall out in something called the G0 phase. And so we've got cells inside our body. You've heard of this, maybe cells of the central nervous system, muscle cells, for example, that never copy themselves during your whole lifetime. They're in what's called the G0 phase, or they're just waiting, and they're not going to make divisions. And so how does a cell know when it's time to divide, and when it should go on and when it shouldn't? The best analogy I can think of is it works kind of like an hourglass clock. And so there are little proteins inside a cell, and as those proteins accumulate throughout the life of the cell, eventually you get a critical number of these proteins at the bottom. And once we have enough of those, then it actually tells the cell to advance to the next stage. And those proteins are called cyclins. And so let's look at what cyclins look like. Cyclin, I'll represent it here. It's simply as a protein. But if we look here at the G1 phase, the S phase, the G2 phase, and mitosis, there's a set of cyclins, or a set of these different types of proteins. And what they're going to do is they're going to build up. And so cyclin A and B are ones that I'm really familiar with. Cyclin A will actually build up during the S phase, and then it'll drop off right as the cell divides, as it goes into this mitotic phase. And so cyclins will actually accumulate. And so those are like the sands through an hourglass. They're going to get more and more and more and more cyclins as a cell goes through the cell cycle. Now, the other chemical that I want to talk about is something called CDK. CDK is simply a cyclin-dependent kinase, and a kinase is simply going to be a chemical that can speed up actions within a cell. And so CDKs are found in all living organisms, and you can actually take CDKs from a yeast and put them in our cell, and they work just as fine. So they show homology through evolution. And so cyclin-dependent kinase if you look at their name, are simply dependent upon cyclin. So I made a little of animation of how that works. And so a typical cell in your body is going to have a bunch of CDKs in it, or cyclin-dependent kinases. And so we could say this is like right here. But throughout the life of the cell is going to start building up and accumulating larger amounts of cyclin. So the amount of cyclin is going to get larger and larger and larger. So eventually what happens is the cyclin is going to fit into the cyclin-dependent kinases. Now we have an activated CDK cyclin complex. What does that mean? We have something, a protein that's able to do things. So now you can think of, we've like mustered this army and now the army is ready to do something. And so what does it do? Well, we're right here, so we're just about to enter into the M phase. So we're just about to do mitosis. And so what these cyclin-dependent kinases do is they act on the cell itself. So they are a specific type of CDK is called the mitosis promoting factor, or MPF. And what that does is when it builds up enough of these cyclin-dependent kinases, they're actually going to work on the cell. So one thing they'll do is they'll actually break apart the nucleus. So we were able to start dividing that cell. Another thing that CDKs will do is they'll actually work on the microtubules that build this spindle. And so all of these together will work on pushing that cell into the mitotic phase, or into this next step of the cell cycle. The neat thing about each of these is that after they've actually done that, they'll actually gobble themselves up, they'll disappear, and then the whole cycle begins over again. Okay, so if we kind of talk big picture about what happens in the cell cycle, a typical cell right here is going to be, let's say this is a typical cell. A typical cell right here is actually going to be 2N. It's diploid. You have one chromosome from mom, one chromosome from dad. So in this case, it's going to be 2N equals 2. It's then going to duplicate itself. So during the S phase, it's going to make copies of itself. And so at this point right here, we'd actually have a 4N cell. It's made copies of that. And at this point, we can either take the path of mitosis or the path of meiosis. And so in the path of mitosis, that'll simply split in half, and now we'll have two 2N cells. And if you look at these two 2N cells, they're exactly the same as that first cell. So this is what's happening to the chromosomes. If we look at that 4N cell, and as it goes into mitosis, it'll actually line up, it'll split in half, and then it'll split in half again. And so what you have is actually N cells. Those are called haploid cells, and we have 4 of those in meiosis. And so the cell cycle will take a diploid cell, make 2 diploid cells in mitosis, or make 4 haploid cells in meiosis. That got a little messy, so let's look at it in a little bit more detail. So mitosis is how we replace cells in the body. So cells that are broken, cells that are broken down, cells that we need to replace, we do that through mitosis. Or when we want to grow, how do we go from a very small organism to a very large organism? It's just making more cells. And so this would be to make it simple, we're going to start with a simple cell. This is going to be a 2N, it's diploid, but 2N equals 2. So this would be that first parent cell. It'll then duplicate all of the DNA. So now we have this characteristic shape. It has 2 sides to it, and this side and that side are copies. That's what happened during the S phase. They'll then meet in the middle. They'll divide in half. So this would be our mitosis phase, and now we have 2 diploid cells. And if you look at those cells, they're exactly the same as that original cell. And each of those are 2N equals 2. So that's mitosis. What happens to these 2N cells? Well, they can enter into the cell cycle again, and it goes over and over and over again. In meiosis, what happens is a little more detailed. Remember, meiosis only deals with making sex cells, or in reproduction. And so in this case, to make it to show you meiosis, I had to increase the number of cells. In this case, or chromosomes, 2N equals 4. In other words, we have 2 chromosomes from mom, 2 chromosomes from dad. So they'll copy themselves. So now we actually have a 4N cell at this point. But since we're doing meiosis, this is where actually crossing over occurs. And so parts of this chromosome will swap places with this chromosome and vice versa. What that gives us is variability in all of the sperm in the egg. They'll divide in half. Now we have 2N cells, and then eventually we'll have N cells equals 2. And so we started with 2N equals 4, now we have N equals 2. Each of these 4 things in a male become a sperm, and in a female, one of them will become an egg, and the other ones will actually form what are called polar bodies. And so what do we get after meiosis? Well, we get sperm and we get egg. And we can then go from these original 2n equals 2 to eventually, or n equals 2 to a 2n equals 4 zygote. And so now we're back to a diploid cell, and that diploid cell could enter into the cell cycle. This is a zygote to make more cells and eventually make sex cells to make the next generation. So that's cell cycle, mitosis, meiosis, and I hope that's helpful.